Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine a disease that, well, quietly takes over 1 million lives every single year.
That's tuberculosis, a global killer, really.
It's staggering. And what makes it so successful, so insidious?
Well, the culprit is this bacterium, Mycobacterium tuberculosis, and its ability to just dodge our immune system. It's quite remarkable. That's what we're diving into today.
Okay, so how does something microscopic manage to outsmart our body's defenses? Our sources point to its outer layer, right? The mycomembrane.
Exactly. Think of it like sophisticated armor.
A biological shield.
Precisely. It's this barrier that helps the bacteria resist attacks. Makes it really persistent.
And really hard to treat.
That's the core of the problem.
Yeah, the treatments, they take months, don't they? And now we're seeing more antibiotic resistance, too.
Which is alarming. It really highlights why we desperately need new approaches, new angles.
Which brings us to the study we're looking at today, published in ACS Infectious Diseases.
That's the one. Researchers have developed a pretty ingenious tool, actually, a chemical probe.
A chemical probe, like a spy molecule.
Sort of. It's designed to investigate a really crucial part of the bacterium's defenses.
Okay, so it targets that mycomembrane shield we mentioned.
Yeah.
But you said it's not just a passive wall.
Not at all. It's quite dynamic. When the bacterium meets our immune cells, especially macrophages, the mycomembrane can actually release molecules.
Molecules that do what?
That actively suppress the immune response. It's like the bacterium is sending out its own calm down signals.
Wow. So the goal of this deep dive, then, is to understand how this new probe works. And what it's telling us about how tuberculosis manages to hide in plain sight, essentially.
Exactly. If we can understand these molecular tricks, maybe we can find new ways to, disable them, new vulnerabilities.
Which could lead to totally new treatments down the line.
That's the hope. Absolutely.
All right, let's get into the nitty-gritty of this study. Mycolic acids. Our sources keep mentioning them. What are they?
Okay, so mycolic acids are these unique fatty acids, very long carbon chains, and they have this characteristic branched structure. They're the main building blocks of that mycomembrane.
The shield components.
Right. Now, they usually exist as part of bigger molecules, glycolipids, but sometimes during remodeling of the membrane, they can be found in free form. We call this free mycolic acid, or fMA.
fMA. Okay. And what does this free form do? How does it help the bacteria hide?
Well, that's key. This fMA is a big part of the slimy stuff, the matrix and tuberculosis biofilms, those bacterial communities that are really tough for drugs to penetrate.
Right, the resistant ones.
Yes. And beyond biofilms, fMA directly helps the bacteria evade our immune response. But how it interacts with our proteins to do that, well, that's been a bit of a black box.
Hence the need for this probe.
Exactly. Which brings us to x-Alk-MA.
The molecular spy. How did they actually build it?
So they synthesized a molecule that's an analog. It closely mimics fMA, but they engineered it with two special features. First, something called a photocross-linking diazerine. Think of it like molecular Velcro that activates with light. It grabs onto nearby proteins and forms a permanent bond.
Activated by light, okay. And the second feature?
That's a clickable alkene. That's like a tiny chemical handle. Researchers can use it to attach fluorescent tags so they can see where the probe went or to pull out the proteins the probe got stuck to.
So it sticks and you can find it. Sounds incredibly precise.
Oh, it is. The chemistry involved, things like asymmetric hydrogenation and diastereous selective alkylation, it's complex stuff, just shows the effort needed to make a good mimic.
Okay, they built this sophisticated mimic, x-Alk-MA, but did it actually act like the real thing inside cells?
Good question. They tested it in macrophage cells grown in the lab the very cells TB infects, and they found that the probe triggered a very similar immune response to what natural mycolic acid does. They checked this using an enzyme immunoassay. So, yes, it seems to be a valid stand-in.
That validation is crucial. So then they use the light activation, the molecular Velcro part.
Exactly. They put the probe into the macrophages, then zapped them with UV light.
Activating the Velcro.
Right. The probe then bonded to whatever proteins it was close to at that moment. Then, using that clickable handle, they attached fluorescent tags.
So they could actually see which proteins got tagged.
Precisely. It's like dusting for the bacterium's fingerprints inside our own immune cells.
That's a great analogy. So whose fingerprints did they find? What protein did x-Alk-MA interact with?
The big discovery was a protein called TREM2. It's a receptor protein found on the surface of these macrophages. They used immunoblotting to identify it, like putting up a molecular wanted poster.
TREM2. Okay. What's known about TREM2? Why is hitting this specific protein significant?
This is where it gets really interesting. TREM2 is actually known to put the brakes on immune cell activation. It dampens the response.
So wait, the mycolic acid mimic binds to a protein that tells immune cells to calm down.
Exactly. It strongly suggests a direct mechanism for how Mycobacterium tuberculosis weakens our immune response. It hijacks TREM2 to send a stop attacking signal.
That is incredibly sneaky, a very clever manipulation. Did they do any controls to be sure?
They did. They used another probe, x-Alk-MA. This one mimics a simpler linear fatty acid without the specific branching of fMA.
And did it bind TREM2?
It showed some interaction, yes, which suggests TREM2 might generally interact with fatty acids to some extent. But critically, the interaction with the proper mimic, x-Alk-MA, seemed stronger.
Meaning the specific structure of the mycolic acid is important for that strong binding.
It really points that way, yes. It's not just any random fat molecule triggering this.
Okay, beyond just binding TREM2, did the probe affect the macrophages in other ways, like the signal they send out?
Yes, they looked at cytokines, those signaling molecules. They found x-Alk-MA caused the cells to produce MCP1.
NCP1.
Monocyte chemoattractant protein 1. It attracts more macrophages. But sometimes the macrophages drawn in by MCP1 are actually ones that Mycobacterium tuberculosis can live inside quite happily. They're permissive.
So it calls in reinforcement that might actually help the bacteria.
Potentially, yes. And importantly, the probe did not trigger the production of TNF-alpha.
Tumor necrosis factor alpha. That's a key pro-inflammatory signal, right?
Yeah.
Usually important for fighting infection.
Very important. So the fact that fMA induces MCP1 but not TNF-alpha, well, it reinforces this idea that fMA is pushing the immune system towards a less aggressive, less effective response. It's actively suppressing the key defensive players.
It's really shaping the battlefield to its advantage. You mentioned another mycomembrane part earlier, Mycolic, TDM. How does fMA compare?
That's a great point. TDM is known to be hyperinflammatory. It really sent the alarm bells, triggers a strong immune reaction.
So the opposite effect of fMA.
Exactly, which highlights how dynamic this mycomembrane is. By changing the balance of components like TDM and fMA, perhaps through remodeling, the bacterium can switch between triggering inflammation and suppressing it.
Like having different messages it can send depending on the situation. Yeah. Fascinating. So pulling back a bit, what are the biggest takeaways, the main implications of this research?
Well, first, it gives us a really valuable new method. This light-activated probe is a powerful tool for actually studying these host pathogen interactions at the molecular level, which has been hard to do.
Better tools mean better understanding. Makes sense. And specifically, finding that TREM2 interaction, what does that clarify about immune evasion?
It shines a bright light on a specific mechanism. We now have evidence that fMA engages with TREM2 on macrophages, and this interaction helps the bacterium dampen the immune response, letting it survive and establish itself.
Okay, so looking ahead, how could this knowledge actually lead to better TB treatments?
That's the ultimate goal. Understanding this interaction could open doors for new strategies, perhaps immunotherapies?
Targeting our own proteins, like TREM2.
Potentially. If you could develop something that blocks fMA from binding to TREM2, maybe you could stop the bacterium from suppressing the immune response. You could, in theory, unleash the host's own immune system to fight the infection more effectively.
A different angle entirely from traditional antibiotics. And you mentioned biofilms earlier, and fMA's role there. Could this probe help tackle drug resistance too?
It could, yes. Since fMA is part of those tough, drug-tolerant biofilms, this probe could be used to figure out exactly how it contributes to biofilm structure and stability.
And maybe find ways to disrupt them.
That would be the hope. Find ways to break down that protective matrix or prevent it from forming, making the bacteria more vulnerable to existing drugs.
Lots of potential then. But like all research, there must be some limitations to keep in mind here.
Oh, definitely. And the researchers are upfront about them. For one, the probe, x-Alk-MA, while it's a good mimic, isn't identical to natural fMA. It has shorter lipid tails, lacks some chemical tweaks found in nature.
So it's a complication.
It's a model, yeah.
Yeah.
Useful one, but we'll need more complex probes eventually to capture the full picture of all the different fMA structures and how they interact.
Understandable. What else?
Well, most of this work was done in cultured cells, cell lines from mice and humans grown in lab dishes.
In vitro studies.
Right. The environment inside a living organism in vivo is way more complex. So the next big step is confirming these findings in animal models of TB infection, seeing if it holds true in a more realistic disease context.
Moving from the dish to the organism.
Mm.
Always a critical step. Anything about the TREM2 binding itself?
Yes, the technique they used, immunoblotting, shows there is an interaction, but it's not great for measuring exactly how strong that interaction is, the binding affinity.
Okay.
So more detailed biochemical tests would be needed to really quantify how tightly fMA binds to TREM2.
And there was something about a linker amide in the probe.
Right. The probe has a chemical linker connecting the mimic part to the Velcro and handle parts. This linker isn't present in natural fMA.
And it might affect how the probe works.
It's possible they acknowledge that. Further studies might need to investigate if that linker influences the interactions they observed. It's just something to be aware of.
Important caveats.
Yeah.
So acknowledging those, what did Ben Swartz, one of the lead researchers, say about the work's value?
He really emphasized the power of using these chemical tools. He said something like, chemical probes can be valuable for discovering and characterizing host pathogen interactions that are otherwise difficult to investigate. It highlights how this approach lets us see things we couldn't see before.
Giving scientists new eyes, essentially. Where do they plan to go next with this research?
They have a few clear directions. They want to make probes with longer lipid chains, different chemical modifications to really map out how the specific structure of fMA relates to its function. Structure-activity relationships.
Fine-tuning the mimic.
Exactly. And they also plan to use this probe more broadly in what's called chemoproteomics. Basically, use it to fish out all the proteins in both the host and the bacteria that fMA might interact with.
So beyond just TREM2.
Potentially, yes, to get a much bigger picture of everything fMA is doing.
That sounds like it could uncover a lot more about TB.
It really could. This study feels like a significant step. By focusing on these free mycolic acids, which maybe haven't gotten as much attention, and building the right tools, they've opened up some really promising avenues.
Yeah, wrapping this up, it really does paint a picture of this constant molecular battle, doesn't it? This arms race between pathogens like TB and our immune system.
It's exactly that, an evolutionary arms race playing out at the tiniest scale. And it shows how understanding these subtle molecular interactions like fMA and TREM2 could genuinely lead us to completely new ways of fighting diseases, maybe moving beyond just trying to kill the bug directly with antibiotics.
Absolutely. Well, if this deep dive into the tactics of Mycobacterium tuberculosis and the clever science trying to uncover them caught your interest, definitely check out the links to the scientific paper and the news article. There's a lot more detail there.
For sure. And maybe it leaves us with a final thought, What other unexpected ways might simple bacterial molecules be messing with our super complex immune system? And what kind of cool new tools will scientists invent next to figure it all out? It really speaks to why we need that constant scientific curiosity, doesn't it?
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.